Hello friends, today we are going to study about protein purification. So first of all, what is the need to purify a protein? So protein purification is needed to study the properties and activities of a particular protein. As we all know, proteins are found in a biological cell. Therefore, to study the activity or the structure of any particular protein, we have to separate or purify the protein from a solution of the proteins found in a biological cell. So first step in protein purification will always be the isolation of protein from a biological cell or tissue. Because the protein is present in a biological cell, therefore we need to disrupt the cell so that the solution of proteins from the cell's interior should come out for us to purify it for its study. So we can go with mechanical disruption by crushing and grinding, followed by filtration and centrifugation to separate the unwanted cellular parts. So we have to mechanically disrupt the cell by crushing or grinding so that the cell will break open and the protein present inside the cell will come out. After breaking the cell, we need to do filtration or centrifugation so that this broken cell part as well as other unwanted cell materials can be separated from the protein mixture. So this is the first step of any protein purification. That is to take the mixture of proteins or the crude protein out from any biological tissue. Now, when we do protein purification or when we extract the crude protein from the biological cell, few conditions are there that we need to keep in mind. What are those? First is the pH. The proteins are pH specific. The proteins function at a particular pH. So we need to control or maintain the pH to maintain the stability of that particular protein. So for that we should always use a buffered solution. Next is temperature. Every protein has its optimum range of temperature for its function. Therefore, we should maintain the temperature so that the stability or the structure of the protein can be maintained. Now, mostly the protein purification is carried out at near 0 degree centigrade, though the proteins get denatured at low temperature also and at high temperature also, but still the high temperatures are more disruptive in nature compared to the low temperature. So we carry out the protein purification nearly at 0 degree centigrade. Next is presence of degradative enzyme. Whenever we break open the cell to extract the crude protein, along with that proteins, many degradative enzymes like proteases, nucleases, they also come out. Now, these proteases can degrade our protein. Therefore, we have to take care of those degradative enzymes so that it should not degrade our desired protein. So, for that, we generally maintain pH and temperature. So these degradative enzymes need to be inactivated, and we can inactivate them by maintaining the pH as well as the temperature, or we can use any blocking agents. Next is adsorption to surfaces. Many proteins get denatured when they come in contact with atmosphere or with the glass. So we should minimize the foaming. So many proteins get disrupted when they come in contact with air or with the glass material or plastic surfaces in which the purification has been carried out. This can be avoided by minimize the foaming. Next is long-term storage. So for the long-term storage, we can use nitrogen. We can store the proteins under nitrogen so that it can slow down the oxidation and microbial contamination. Now, how proteins are quantified? Before the protein purification, we need to quantify the protein. That means we need to quantify the crude extract so that we can know how much amount of proteins are present. Correct? And after each step of purification also, we need to quantify the protein so that we can understand how much amount of our desired protein is present in the final sample. So how we can quantify the protein? So for that, the first is the protein an enzyme? If it is an enzyme, then we can quantify using the enzymatic reaction. If the protein is an enzyme, so this enzyme will act on a substrate and will form an enzyme substrate complex or a protein substrate complex, which will convert into product, giving the enzyme back. So as we all know, the rate of product formation is proportional to the amount of enzyme present. Therefore, depending upon the amount of the product obtained and the rate of product formation, we can easily calculate the amount of the enzyme if our protein is an enzyme. Now for that, the product should be in the form of quantifiable, observable product. Now, if the product is not observable, we cannot observe or quantify the product. In that case, what we can do, we can convert the product again. This is our enzyme or the protein which we need to quantify. If this protein or this enzyme is giving rise to a product which is not observable, then we can convert the product into some another product which can be quantified easily or it is observable. So this how, if the protein is an enzyme, then we can use the enzymatic reaction to quantify the protein because the rate of product formation is proportional to the amount of enzyme present. Now, what if the protein is not an enzyme? So for that, different assays are present. First is immunoassay. What do we mean by immunoassay? Immunoassay means if our protein is an antibody, then we can react the antibody with their respective antigen. Now, this antibody and antigen interactions can be easily quantifiable because it is observable. So this is known as immunoassay if our protein is an antibody. Next is radioimmunoassay. If our protein is an antigen, then what we can do, we can use the corresponding antibody and we can flood our antigen as well as the radio-labeled antigen over this antibody. So what will happen? Our antigen and radio-labeled antigen will compete with each other to bind to the antibody. If the concentration of antigen is more, so more of antigen will bind with the antibody and less of radio-labeled antigen will bind. If the concentration of antigen is less, in that case, less antigen will bind with the antibody and more of radio-labeled antigen will bind with the antibody. As because this radio-labeled antigen is observable and detectable, so based on the reading of radio-labeled antigen, we can easily quantify the amount of antigens present in a sample. Next is an enzyme-linked immunosorbent assay. Enzyme-linked means here we'll use one enzyme to get our observable product. Now, what is happening? This antigen, this is direct ELISA. The antigen is already bound to a plate. With this will bound primary antibody. This antibody is linked with the enzyme which can convert the substrate into observable product. In case of indirect ELISA, the antigen is bound with the plate. Over that antigen, primary antibody is bound, but this primary antibody cannot convert the substrate into product. So we need a secondary antibody. This secondary antibody is linked with an enzyme and can convert substrate into observable product. So by seeing the observable product, we can quantify the amount of antigen present in the sample. Sandwich ELISA, in this will bind antibody first on a plate, then we will flush our sample antigen over the antibody. If our antigen is present, it will bind to the antibody to which primary antibody is bound. And again, a secondary antibody is bound, which is linked with an enzyme to convert the substrate into an observable product. And by seeing the result of the observable product, we can detect the amount of antigen present in the sample. In case of competitive ELISA, we have our antigen and we have some inhibitory antigens. So both will compete to bind with the same antibody. If the antibody is bind with our enzyme, it will convert the substrate into product. If the antibody binds with an inhibitory antigen, then it cannot convert the substrate into product. So depending upon the concentration, if the concentration of antigen is more, so we'll get more product. If the concentration of antigen is less, we'll get less product because in that case, the antibody will bind more with the inhibitory antigen. So this how on the basis of the product, as we can observe the product, as we can measure the product, so depending upon that, we can quantify the amount of protein present in a sample. Now, how we can determine protein concentration? So to determine protein concentration, we need a method called spectroscopy, which is based on Beer-Lambert law. What is Beer-Lambert law? The absorbance depends upon the concentration and the length of light path. So absorbance, which we can determine with the intensity of incident light at a given wavelength divided by intensity of transmitted light at a given wavelength. Correct? So Beer-Lambert law works on a particular wavelength. On the particular wavelength, the absorbance will depend on ECL, that is, C is the concentration and L is the length of the part and E is the absorptivity. So now the value of E will vary wavelength and wavelength is denoted by lambda. Correct? Because Beer-Lambert law depends upon a particular wavelength. So if we vary the wavelength, the absorptivity will vary. Okay, so now if we plot absorbance or absorptivity with the lambda, that is a wavelength, we can get an absorption spectrum. Now all the polypeptides, or we can say the proteins or amino acid, they absorb in the range of ultraviolet. because they have an aromatic side chain, that is phenylalanine, tryptophan, and tyrosine. Because of this, a polypeptide or protein will absorb the wavelength of ultraviolet region. So we can get a graph. The graph is showing of phenylalanine, tyrosine, and tryptophan. At 260 nanometer, the absorptivity is more. Phenylalanine, this is what tyrosine, and this is what tryptophan. So tryptophan will absorb more than tyrosine and then phenylalanine. Okay. So you can remember by this sequence. Tryptophan absorbs more than tyrosine and then phenylalanine at 260 nanometer. Okay, so we can determine the protein concentration by spectroscopic method. Now, one question from my end for you all, that is, which peptide has greater absorbance at 280 nanometer? Two peptides are given, peptide A and peptide B, having a particular set of amino acids. Now you have to tell me which peptide will absorb more at 280 nanometer. How you can get? You have to check the amino acids present in each peptide. If you check that and if you match that with the spectroscopic slide, you can easily tell me that which peptide will have a greater absorbance. Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.